Hi, welcome to Chapter 10, Part A. Today we're going to talk about the nervous system. In this section, we're going to explore the structure, function, and complexities of the nervous system. This is an important concept to understand because you will be talking about this again in future courses, especially in pain management when we're talking about anesthetic and how that affects the nervous system. Okay. So today our objectives include understanding the organs and divisions of the nervous system, exploring the major types of cells and their functions. We'll also discuss the structure of the reflex arc and the mechanism behind the nerve impulse transmission. Okay, so organs and divisions of the nervous system. So what are the two principal divisions of the nervous system? It's going to be the central nervous system and the peripheral nervous system. So those are the two principal divisions. Okay. The nervous system consists of the central nervous system, which includes the brain and spinal cord, the peripheral nervous system, which is encompassing all the nerves. And then we do have the autonomic nervous system, which is the ANS, and that's responsible for all of our involuntary actions. So here is a chart that kind of breaks down the central and the peripheral nervous system. Okay, so the central nervous system is very simple, brain, spinal cord, and it's integrative and it's the control center. So the peripheral nervous system is your cranial nerves, your spinal nerves, and the communication line between the CNS and the rest of the body. And it's broken down into sensory and motor, and then motor is broken down even more into somatic nervous system and the autonomic nervous system. And the autonomic nervous system is broken down even more into the sympathetic and the parasympathetic divisions. So there's cells of the nervous system. What are the two types of cells found in the nervous system? So that's going to be neurons or nerve cells and glia, which is specialized connective tissue cells. So neurons, they consist of three parts. So you have your cell body of the neuron. So that's the main part, houses the nucleus. You have dendrites, which is the main receptive input region of the neuron, and in it conducts impulses to cell bodies of the neuron. And then you have your axon, which is elongated projection that conducts the impulses away from the cell body of the neuron. So axons have occasional branches along their length called axon collaterals. And they have branches on them at the end, which is called terminal branches, and they have like knob-like endings called axon terminals. All right, so when we're looking at this, what is the direction of neural transmission from sensory neurons and motor neurons? So sensory neurons, they transmit impulses to the spinal cord and brain. Motor neurons transmit impulses away from the brain and spinal cord. And then what are interneurons? So interneurons conduct impulses from sensory neurons to motor neurons. So they're also called central or connecting neurons. So in general, the nervous system, it includes neurons, which are responsible for transmission signals, glia cells, which provide support and protection, and neurons have a cell body, dendrites for input, and axon for output. So here is what that cell looks like. Okay, so you can see the cell body that holds the nucleus, and then attached to the cell body is the dendrites. That's what receives input signals. Then you have your axon, which is this long sheath coming from the cell body that sends signals away from the cell. So this diagram shows the key parts of the neuron. The dendrites receive signals, the axon carries them away, and we're going to talk about those sheaths that are around the axon and what those are called. They're called myelin sheaths, and how they are used to speed up the process of transmission. All right, so when we look at our neurons, we've talked a little bit about this, but we have sensory neurons, motor neurons, and interneurons. So again, sensory neurons conduct impulses to the spinal cord and brain. They're also called afferent neurons. Motor neurons conduct impulses away from the brain and spinal cord to the muscles and glands, and those are efferent neurons. And then interneurons conduct impulses from sensory neurons to motor neurons. And then again, they're also called central or connecting neurons. So motor neurons conduct impulses in only two kinds of tissue. That's in muscle and glandular epithelial tissue. Okay, so the myelin sheath, that isolates the axons and speeds up nerve impulses. So myelinated fibers transmit signals faster than unmyelinated fibers. So if we go back and look here, this these yellow bubbles around the axon, those are myelinated sheaths. So what happens is, why it goes faster is the signal is being transmitted, it hits this sheath, and it has to jump around it. And so it jumps, 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 jumps from called nodes of Ranvier. So it jumps from these nodes of Ranvier. So it goes faster. If it did not have these myelinated sheaths, if it didn't have these yellow bubbles, it would be unmyelinated, which means the signal has to travel down the axon one action potential at a time, which we're going to talk about. So it's a little bit slower of transmission. Instead of going jump, 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 it just has to go slowly down the entire axon. All right, so classification of neurons. They're classified by classified structurally. So you have multipolar neurons, which have many processes. You have bipolar neurons that have two, and unipolar neurons, which have a single T-like process. Here is pictures of what those look like and where they can be found. So this is a really good chart to look at and compare the difference. So another cell that we've mentioned is the glia, which is the neuroglia. So These are support cells, and they bring cells of the nervous tissue together structurally and functionally. So the function of glia cells is glia is Greek for glue. So it holds the functioning neurons together, protects them, and regulates neuron function. There are three types. You have the astrocytes, which are relatively large star-shaped cells that attach to neurons and small blood vessels to hold these structures close to each other. Then you have microglia, which is usually remains stationary, but in inflammation or degeneration of the brain, they enlarge and they can move about and act as microbe eating scavengers. And then you have the oligodendrocytes, which help hold nerve fibers together and also produce the fatty myelin sheath. So myelin is a fatty white substance around those cells, and uh, glial cells, they help to support the neurons. You can think of astrocytes that help anchor the neurons. Microglia act as immune cells, and the oligodendrocytes create myelin sheaths in the CNS. And Schwann cells do this in the PNS, which we'll talk about. And here is a picture of what those three different cell types look like. All right, so nerves, you have your nerve bundle of the peripheral axons. Your tract, you have a bundle of central axons. White matter is your brain or cord tissue, and it's composed primarily of myelinated axon or tracts. And gray matter is brain or cord tissue composed primarily of cell bodies and unmyelinated fibers. Okay, so what is white matter composed of? It's nerve fibers that usually have a myelin sheath, and myelin is white. So that's what causes the white color of the matter. So what is gray matter composed of? It's tissue composed of cell bodies and unmyelinated axons and dendrites. And it's called gray matter because of its characteristic gray appearance. Okay, so here is what that looks like broken down. So this illustrates the nerve. Each nerve contains axons, and they're bundled into fascicles. And a connective tissue, or the epineuron, wraps the entire nerve. Perineurium surrounds each fascicle, and the endoneurium surrounds each axon. Okay, so here we have one single axon. That endoneurium is what surrounds that axon. Okay, then when you have a bundle of axons, it's called a fascicle, and the sheath that surrounds that is called the perineurium. And then when you have your whole nerve cell here, which contains multiple fascicles, the whole entire nerve is surrounded by the epineurum. Okay, so again, we're talking about the endoneurium, perineurium, and epineurum. How would you describe the coverings that surround an axon? Okay, So the axon is surrounded by the thin wrapping of fibrous connective tissue called the endothelium. Okay. They are then bundled together in groups called the fascicles and wrapped in an outer layer of perineurium. Then the whole nerve is surrounded by the tough sheath called the epineurium. So nerve impulses, where does the impulse conduction originate? So normally it starts in receptors and the beginnings of dendrites of sensory neurons. And the end of the sensory neurons, axon synapses, first with the interneuron before chemical signals are sent across a second synapses, which we're going to talk about all these synapses later in this chapter. But that results in conduction through the motor neurons. So for example, application of an irritating stimulus to the skin of the thigh initiates a 3-neuron reflex response that causes contraction of the muscle to pull the leg away from that irritant. Okay. So basically, nerve impulses or action potentials are electrical signals that travel along the neurons, and when they're triggered by stimulate, they propagate down the axon to cause a reaction, like your muscles pulling away from that stimulus. All right, so reflex arcs. A reflex arc is the simplest type of neuron pathway. So nerve impulses or action potentials are conducted from receptors to effectors over neural pathways or reflex arcs. Okay. So, for example, contraction of a muscle or secretion by a gland. They're the simplest. Reflex arcs are 2 neuron arcs, consisting of only 2 types of neurons, which is sensory neurons and motor neurons. And then what is a 3-neuron arc? So that consists of 3 different types of neurons: sensory neurons, motor neurons, and interneurons. So the reflex arcs involves a direct pathway from sensory neurons to motor neurons, and it enables quick responses like muscle contraction or gland secretion. All right, so this is the patellar reflex, right? When they hit your knee with that little instrument there, and it causes your knee-jerk reaction without your response, right? So this figure shows the neural pathway involved in the knee-jerk patellar reflex. So a reflex arc is like a one-way street because the impulses can be conducted in only one direction. And with the patellar reflex, it's an example of a simple 2-neuron reflex arc, and it demonstrates how sensory and motor pathways work together. All right, so when we look at this picture here, it shows the mechanism of a nerve impulse or a voltmeter on the right. It shows how the charge the charge difference across the membrane and how it fluctuates the balance of positive ion changes. And we're going to watch a video on this, how this works as well, that might explain it a little bit easier. But nerve impulses, they're basically electrical signals caused by changes in the membrane potential, starting with depolarization and ending with repolarization. And we're going to be talking about this in depth. And this is what you really need to understand, especially when it comes to pain management and giving anesthetic. All right, so mechanisms of the nerve impulse. So when your nerves are at rest, your neuron's membrane is slightly positive on the outside, which means it's polarized. And from a slightly excess of sodium on the outside of the neuron. On the inside of the neuron is potassium, which makes it more negative. So a stimulus that triggers the opening of sodium channels in the plasma membrane of the neuron will allow neurons from outside the neuron inside. So different types of stimulates that could initiate nerve impulses are pressure, temperature, and chemical changes. All right, so conduction of the nerve impulses. Okay, so when a nerve is conducting impulses along the neural pathway, what happens is there has to be a change in the positive to negative frequency across that membrane. So you get inward movement of the positive sodium ions that are outside the neuron. They depolarize across the membrane by making it inside more positive than what is outside at this point. And this depolarizes the nerve impulse. It's called an action potential. And when this happens, the stimulated section of the membrane immediately repolarizes, meaning some of the potassium inside the inside the nerve is going to go outside of the nerve, and it's trying to repolarize and get back to the neutral balance from a positive negative. So when it does this, by the time that the depolarization has already triggered the next section of the membrane, and then that's how it sends that signal down. So it starts at one part and then it just depolarizes all the way down the nerve, and that's how it pushes that signal down the nerve. So why are nerve impulses called action potentials? And it's because each one is a difference in charge called electrical potential that triggers an action by the cell. Here's an illustration of how this works as well. You can see on the outside of the neuron is the positive sodium, and on the inside is the negative potassium. So when you have an exchange of positive sodium entering into the neuron and you have some negative potassium exiting for the depolarization, it causes the action potential and that electrical signal to go down the length of the neuron here. So it starts here and then it keeps going all the way down. Now, when you have the myelinated sheaths, what it does is it skips the part that's myelinated, right? So it will jump from um, node of Ranier to node of Ranier. Now, if it wasn't myelinated, then it would just keep going down. As you see here, it would follow down that axon. Okay, so here we see this figure, it shows the conduction of the nerve impulses in an unmyelinated fiber, with the action potential shown in the yellow, and with a continuous self-propagating wave of electrical disturbance. The dark blue area of the recovery during repolarization cannot be re-stimulated, so it prevents backward conduction. So if we see this dark blue behind the yellow, it doesn't matter what trigger or stimuli is given, this area cannot be triggered again, because we don't want that signal being confused and going back and forth and back and forth. We just want that signal to go in one direction. So in the myelinated fiber, the action potential jumps around the insulating myelin in a rapid type of conduction. That is called saltatory conduction. So when it's jumping from node of Ranvier to node of Ranvier, that's called saltatory conduction. So again, saltatory conduction is a much faster type of impulse travel and occurs on the myelinated neurons. So, and saltatory conduction allows the impulses to jump between the nodes of Ranvier, and it speeds up that signal transmission in the myelinated neurons. So synapses, there's 3 different structures that make up a synapse: a synaptic knob, a neurotransmitter, and a synaptic cleft. And you also have your plasma membrane. Your neurotransmitters bind to specific receptor molecules in the membrane, and it helps to open those ion channels and to create that stimulating impulse by the membrane. So how does a nerve impulse travel from one neuron to another? And that is through the synapse via the neurotransmitter. All right. Okay, so what is a neurotransmitter and how many are there? So neurotransmitters are chemicals by which neurons communicate. There are at least 30 different compounds that have been identified as neurotransmitters. So some of those are listed on this slide here, but there are more than what is listed, at least 30. So you have your acetylcholine, which is released at some of the synapses in the spinal cord and at neuromuscular junctions. Your norepinephrine, dopamine, and serotonin belong to a group of compounds called catecholamines, which may play a role in sleep, motor function, mood, and pleasure recognition. And then you have 2 morphine-like neurotransmitters called endorphins and enkephalins, and they're natural painkillers. So this is what a synapse looks like. All right, so you have your synaptic knob right here, or it's also called an axon terminal of the presynaptic neuron. You have your plasma membrane of the postsynaptic neuron, which is this right here. So your plasma membrane, and then a synaptic cleft. So this is the space between the membrane and the synaptic bulb, and that is called the synaptic cleft. So you have in the boxes steps that summarize the process of synaptic transmission of a nerve signal. So on the arrival of an action potential at the synaptic knob, neurotransmitters, right, which are in these vesicles. These neurotransmitter molecules are released from the vesicles in the knob into the synaptic cleft here. And then the combining of the neurotransmitter and the receptor molecules in the plasma membrane of the postsynaptic neurons opens ion channels. So these right here are your ion channels that get opened, and it initiates the impulse conduction in the postsynaptic neuron. All right, so on to the next one. We have our membrane potentials. Like all cells, neurons have resting membrane potential. And neurons can rapidly change resting membrane potential. So to understand how this works, we want to understand the basic principles of electricity. So opposite charges are attracted to each other. So when we have a voltage, that is a measure of potential energy. So we're going to watch a video here later as well that compares this to like a battery, where you have potential energy in that battery and it can be measured. And this is called the potential difference, or the potential. And that charge is the difference across the membrane plasma that results in that potential. So a current is the flow of the electrical charge, or the ions, between the 2 points. Resistance is any hindrance to the charge flow. Insulator is a substance with high electrical resistance. So if you think of electricity, an insulator might be plastic, right? It does not conduct electricity easily. A conductor is a substance with low electrical resistance, and that could be like copper or metal, where it conducts that electrical source very easily. All right, so your role of your membrane to ion channels, just recall that plasma membranes are peppered with a variety of proteins that can act on those ion channels. And the channel is selected as to the type of ion that is allowed to pass, right? So a sodium ion channel only allows sodium to pass, where a potassium, K is potassium. The potassium ion channel only allows potassium to pass. The sodium ion channel only allows sodium to pass, okay? Large proteins serve as selective membrane ion channels, which enables two main types of ion channels, which is non-gated, which is also called leakage channels, which are always open, and then gated channels, which part of the protein changes shape to open and close that channel. So there are three main types of gated channels. You have chemically gated, which is also called a ligand gated channel. It only opens when a specific chemical binds to that gate. You have a voltage gated channel, which open and close in response to changes in the membrane potential. And then you have mechanically gated channels, which open and close in response to physical deformation of the receptors. All right, so when gated channels are open, the ions diffuse quickly through those channels along chemical concentration gradients from higher to lower concentration, and went along electrical gradients, which is towards the opposite electrical charge. So positive to negative, negative to positive. And then electrochemical gradient, which is electrical and chemical combined. Ions flow creates an electrical current, and the voltage changes across the membrane. Our resting membrane potential, is approximately negative 70 millivolts inside of the neuron. The resting membrane potential is the electrical charge difference across a neuron's membrane when it's not actively transmitting a signal, and it's maintained by ion channels and the sodium-potassium pump. All right, changing the resting membrane potential. So membrane potential changes when concentration of the ions cross the membrane change. And then changes are produced two types of signals. So you have graded potentials, which are incoming signals that operate over short distances. Okay, so incoming signal via a dendrite towards the cell body. And it's only in a very short, limited area. Action potentials, however, go a long distance across those axons. All right, so changing the resting membrane potential, terms that would describe this is depolarization, which is a decrease in the membrane potential, repolarization, restoration of the resting membrane potential after the depolarization event, and then hyperpolarization. So that increases the membrane potential, and the inside of the membrane becomes more negative than the resting membrane potential. And the probability of producing that impulse decreases during that phase. So again, graded potentials are changes in the membrane potential. They vary in strength and do not follow the all or none principle. These occur in the dendrites and the cell body, right? So very limited, short distance. And unlike action potentials, graded potentials are short-lived, localized, and decrease in strength over distance. Action potentials are signals sent over long distances. They can only occur in muscles and axons of neurons. They do not decay over distance like graded potentials do. And in neurons, they are referred to as nerve impulses. So there's four main steps in generating an action potential. So an action potential is a rapid change in the membrane potential that propagates down the axon, and it involves depolarization, repolarization, and then sometimes the hyperpolarization. So in the resting state, all the sodium and potassium channels are closed. Only leakage channels, which are always open, maintain the balance between sodium and potassium. And then in your depolarization, it occurs when the sodium channels are open, and it allows the sodium ions to enter the cell, reducing the membrane potential towards zero. So depolarization. So the depolarizing local currents opens voltage gated sodium channels. The sodium rushes into the cell. Then the sodium causes more depolarization, which opens even more sodium channels, which allows more sodium in, and it just has this chain reaction all the way down the axon. So at the threshold of negative 55 millivolts, positive feedback causes opening of all of the sodium channels, and that results in a large action potential spike, lots of sodium crossing over in the membrane, and then that polarity jumps up to 30 millivolts. In the repolarization, sodium channels are inactivated and potassium channels are open. So then this allows it stops the sodium channel from coming into the nerve, and it allows the potassium to exit the nerve. So it exits this down the cell, down the electrical gradient to repolarize, rebalance that membrane, and it gets down into the -70 millivolts again. So repolarization restores the resting membrane potential, and it happens when sodium channels are closed, potassium channels are open, and allows potassium ions to exit the cell. During hyperpolarization, the membrane potential becomes more negative than the resting potential due to continued potassium ion overflow. So it kind of overdoes it a little bit. And this is how we can see on the chart how it goes from -70. It's during that depolarization where the sodium is coming into the nerve cell, into the axon, it jumps up to +30. And then with repolarization, closes the sodium cells, open up potassium to help get that back to where it's supposed to go. It actually goes down below -70 in that hyperpolarization before it gets back to that -70 threshold. So for an action potential to occur, the membrane must reach a specific threshold, which is typically around -55 millivolts, and that triggers the opening of the voltage-gated sodium channels. And action potentials follow an all or none principle. So they're either occur fully or not at all, and it depends on whether that threshold is reached. All right, an action potential is propagated along the axon by depolarizing adjacent segments of the membrane and ensures the signal reaches its destination. All right, so here we have our non-myelinated, which this is when the sodium influx causes that depolarization in one area, and then that depolarization causes it, then it causes depolarization in the next adjacent area, and it goes all the way down, causing that cascading train reaction all the way down the axon. And once it's initiated, the action potential is self-propagating and it continues along the axon in a consistent philosophy. And it only occurs in one direction, which is in the forward direction. With myelinated, most sodium channels are concentrated only in those gaps between the Schwann cells, which is the nodes of Ranvier. In some process overall, however, the action potential, it moves from one gap to the next gap instead of along the entire axon. So like we said before, it has like a jumping effect. It's jumping over those myelinated areas. So continuous conduction occurs in unmyelinated axons, while saltatory conduction happens in myelinated axon, which allows those impulses to jump between the nodes of Ranvier, and it speeds up that transmission. And this is what an example of what each of those look like. So the refractory period is that time which the neuron cannot be triggered by another action potential. So you have two types. You have your absolute refractory period. That's the time from the opening of the sodium channels until the resetting of the channels, and it ensures that the action potential is an all or none event. And in the absolute refractory period, it doesn't matter how much stimulus, no amount of stimulus will start another action potential. In a relative refractory period, it allows It does follow the absolute refractory period. Most of the sodium channels have returned to their resting state. Some of the potassium channels are still open and repolarization is occurring. You there is the potential for another action potential, but it's only really exceptionally strong stimulus could stimulate a new action potential. And here's has kind of same chart that we've seen. And here's a little video that really explains what we've been talking about in more detail and an easy way to understand.
What if everything you did, and thought, and felt could be communicated by pushing a button? It'd be like using the world's simplest app, one that just sends out a little ping, always at the same volume and length, to communicate everything from "it sure is cold in here" to "I love churros" to "boy, I sure would like to breathe sometime soon." Well, that is actually exactly how your neurons send all the impulses responsible for every one of your actions, thoughts, and emotions. When a neuron is stimulated enough, it fires an electrical impulse that zips down its axon to its neighboring neurons. But they've only got one signal that they can send, and it only transmits at one uniform strength and speed. What they can vary is the frequency or number of pulses, like this is distinct from this. And your brain can translate these signals, reading them like binary code, organizing them by location, sensation, magnitude, and importance, so that you know the difference between "turn up the thermostat" and "oh my gosh, I'm on fire." That buzz, that nerve impulse, is called the action potential. It's one of the most fundamental aspects of anatomy and physiology, and really life in general. It's happening inside of you right now, and we want to make sure that you understand what all that buzz is about. Before we delve into how neurons communicate, we gotta first understand a little bit of our old friend electricity. Basically, think of your body as a sack of batteries. No, I mean, you don't look like a sack of batteries. I'm just saying that your body as a whole is electrically neutral with equal amounts of positive and negative charges floating around, but certain areas are more positively or negatively charged than others. And because opposite charges attract, we need barriers or membranes to keep positive and negative charges separate until we're ready to use the energy that their attraction creates. In other words, we keep them separated to build potential. A battery, just sitting on its own, has both a positive and a negative end, and the potential to release energy, but it doesn't do anything until it's hooked up to a flashlight or a phone or a kid's toy that lets those charges move toward each other, on the way converting electricity into light or sound or children's laughter. In much the same way, each neuron in your body is like its own little battery with its own separated charges. It just needs an event to trigger the action that brings those charges together. If you're thinking that this sounds more like engineering than anatomy, that might not be a bad thing. It might even help to think of your neurons in the same terms an electrician might use. Voltage, for example, is the measure of potential energy generated by separated charges. It's measured in volts, but in the case of your body, we use millivolts because it's a pretty small amount. In a cell, we refer to this difference in charge as the membrane potential. The bigger the difference between the positive and negative areas, the higher the voltage and the larger the potential. And just like there's voltage in your body, there's also current, the flow of electricity from one point to another. The amount of charge in a current is related both to its voltage and its resistance. Resistance is just whatever's getting in the way of the current. Something with a high resistance is an insulator, like plastic, and something with a low resistance is a conductor, like metal. Now, when we talk about these concepts in terms of you, we're typically talking about how currents indicate the flow of positively or negatively charged ions across the resistance of your cell's membranes. And again, these membranes separate the charges, so they're what provide the potential to convert the electricity into something useful. Okay, now that we got Electricity 101 down, let's see how it works inside your nervous system. A resting neuron is like a battery just sitting in that sack that is you. When it's just sitting there, it's more negative on the inside of the cell relative to the extracellular space around it. The difference is known as the neuron's resting membrane potential, and it sits at around negative 70 millivolts. Where do those charges come from? Outside of a resting neuron, there's a bunch of positive sodium ions floating around, just lingering outside the membrane. Inside, the neuron holds potassium ions that are positive as well, but they're mingled with bigger negatively charged proteins. And since since there are more sodium ions outside than there are potassium ions inside, the cell's interior has an overall negative charge. When a neuron has a negative membrane potential like this, it is said to be polarized. Now, these ions didn't just show up in this arrangement on their own. This is all orchestrated by one of the most important bits of machinery in your nervous system, the sodium-potassium pump. This little protein straddles the membrane of the neuron, and there are tons of them all along the axon. For every 2 potassium ions it pumps into the cell, it pumps out 3 sodium ions. This creates a difference in the concentration of sodium and potassium, and a difference in charges, making it more positive outside the neuron. This difference is an electrochemical gradient, and you probably know enough about biology by now to know that nature hates gradients. It wants to even out all of those inequalities in concentration and in charge to restore balance. But the only way to even out that gradient is for the ions to pass across the membrane. Thankfully, the sodium-potassium pump isn't the only way in or out of the cell. The membrane is also riddled with ion channels, large proteins that can provide safe passage across the membrane when their respective gates are open. And these gates open and close for different reasons depending on their structure and purpose. Most are voltage-gated channels, which open at certain membrane potentials and close at others. For example, sodium channels in your neurons like to open around -55 millivolts. But some others are ligand-gated channels. They only open up when a specific neurotransmitter, like serotonin or a hormone, latches onto it. Then we also have mechanically gated channels, which open in response to physically stretching the membrane. In any case, when the gates do open, ions quickly diffuse across that membrane down their electrochemical gradient, evening out the concentrations and running away from other positively charged ions. This movement of ions is the key to all electrical events in neurons, and thus is the force behind every single thing we think, do, and feel. Of course, not all of your body's electrical responses are the same, and neither are the flows of ions going in and out of your neurons. If only a few channels open and only a bit of sodium enters the cell, that causes just a little change in the membrane potential in a localized part of the cell. This is called a graded potential. But in order to send long-distance signals all the way along an axon, you need a bigger change, one big enough to trigger those voltage-gated channels. That is an action potential. And your best bet for making that happen is to depolarize that resting neuron. I mean, cause a big enough change in its membrane potential that it'll trigger the voltage-gated channels to open. It all starts with your neuron sitting there at resting state. All of the ion channels are closed and the inner voltage is resting at -70 millivolts. And then something happens. Some environmental stimulus occurs, say like a spider brushes up against a tiny hair on your knee, triggering those sodium channels to open, increasing the charge inside the membrane. Now, the stimulus and the resulting change have to be strong enough to cross a threshold for the true action potential to kick in, and that threshold is about -55 millivolts. Remember that number, because this is an all-or-nothing phenomenon. If the stimulus is too weak and the change doesn't hit that level, it's like a false alarm. The neuron just returns to its resting state. But kind of like Doc Brown hitting 1.21 gigawatts in the DeLorean, once it hits that threshold, you're not going to travel in time, but you are going to see some serious action potential. At that threshold, the voltage-gated sodium channels open, and there are tons of these, so all of the positive sodium ions rush in, making the cell massively depolarized. So much so that it actually goes positive, up to about +40 millivolts. This is action potential in action. It's really just a temporary reversal of a membrane potential, a brief depolarization caused by changes in currents. And unlike graded potentials, which are small and localized, an action potential kicks off a biological chain reaction which sends that electrical signal down the axon. Because each of your neurons has lots of voltage-gated sodium channels, so when a few in one area open, that local current is strong enough to change the voltage around them. And that triggers their neighbors, which triggers the voltage around them, and so on down the line. As soon as all that's underway, the process of repolarization kicks in. This time the voltage-gated potassium ion channels open up, letting those potassium ions flow out in an attempt to rebalance the charges. If anything, it goes too far at first, and the membrane briefly goes through hyperpolarization. Its voltage drops to -75 or so millivolts before all of the gates close and the sodium-potassium pumps take over and bring things back to their resting level. Now, when part of an axon is in the middle of all this and its ion channels are open, it can't respond to any other stimulus, no matter how strong. This is called the refractory period, and it's there to help prevent signals from traveling in both directions down the axon at once. So that is the surprisingly simple app that your nervous system uses to let you experience the world. And because the voltages in this process are always pretty much the same, the initial threshold around -55 millivolts and the peak depolarization at 40 millivolts, your neurons only communicate in a buzz. It doesn't matter if it's a paper cut or a stab wound, the strength of that action potential is always the same. What does change is the frequency of the buzz. A weak stimulus tends to trigger less frequent action potentials, and that includes if the stimulus is coming from you, like your brain telling your muscles to perform some task. If I need to do something delicate, like pick up an egg, the signal is low frequency. But a more intense signal, like trying to crush a can, increases the frequency of those action potentials to tell your muscles to contract harder, and the message turns into something that you can't ignore. Action potentials also vary by speed or conduction velocity. They're fastest in pathways that govern things like reflexes, for example, but they're slower in places like your glands, guts, and blood vessels. And the factor that affects a neuron's transmission speed the most is whether there's a myelin sheath on its axon. Axons coated in insulating myelin conduct impulses faster than non-myelinated ones, partly because instead of just triggering one channel at a time in a chain reaction, a current can effectively leap from one gap in the myelin to the next. These little gaps are the delightfully named nodes of Ranvier, and this kind of propagation is known as saltatory conduction, from the Latin word for leaping. But what happens when an action potential hits the end of its axon and is ready to do more than leap and jump all the way to another neuron? That you will find out next time. Today, you learned how your body is kind of like a big bag of batteries, and how ion channels in your neurons regulate this electrochemistry to create an action potential from resting state to depolarization to repolarization, and a brief bout of hyperpolarization. Thanks for watching, especially to all of our subbable subscribers who make Crash Course possible for themselves and for everyone else. To find out how you can become a supporter, just go to
All right, so hopefully that was helpful to explain what we were just talking about there in an interactive visual way. All right, now we're moving on to the synapse. Excuse me. Neurons are functionally connected by synapses, which are junctions that mediate information transfer, right? So you have a bunch of different neuron cells. How do you get information from one neuron cell to the next neuron cell? So a synapse is when one neuron communicates with another or with an effector cells. And chemical and electrical synapses play distinct roles in this process. All right, so in chemical synapses, they involve a neurotransmitter crossing the synaptic cleft to bind to receptors on the postsynaptic neuron, which initiates a response. So here you have electrical impulse changes to chemical messages across the synapse, then back into an electrical process, right? So you have these neurotransmitters here. They're released into the synaptic cleft. These chemicals then attach here to the receptors, and then that allows ions in to start that electrical impulse again. So there's 6 steps to transfer across a chemical synapse. So first, the action potential arrives at the presynaptic axon terminal. Then you have that voltage gated channel that opens up the axon terminals. And then that synaptic vessel, then it causes the synaptic vesicles inside of there to release by exocytosis. The neurotransmission diffuses across the synaptic cleft, but binds to the receptors on the postsynaptic membrane. That causes the opening of those ion channels and results in that graded potential right in that localized area. And then the neurotransmitter effects are terminated by the reuptake or diffusion. So again, here's another picture we've seen this picture several times, how these are released into the synaptic cleft, attached to the transmitter, open that channel up, and then it causes that electrical pulse. So electrical synapses are less common than the chemical synapses. This is when the neurons are electrically coupled, and communication's very rapid, but may be unidirectional or bidirectional. So electrical synapses allow the ions to flow directly between the neurons and enables the rapid and synchronized communication. So neurotransmitters are the chemical messengers of the nervous system, such as acetylcholine and dopamine, and they each have their specific roles and their effects. There are 50 or more neurotransmitters that have been identified. Most neurons make 2 or more neurotransmitters, and they are classified by their chemical structure and their function. So here's how we can classify by their chemical structure. So you can look over this chart. It's in the PowerPoint. Look over that more closely. When we classify them by function, neurotransmitters, they exhibit a lot of diversity of functions. It's determined by the receptor to which it can bind to, and it's classified in 2 groups. So effects, which is excitatory versus inhibitory, and then actions, which is direct versus indirect. So effects, which is excitatory or inhibitory, this is when neurotransmitters when their effects are excitatory, this is when it depolarizing happens. And when they're inhibitory, this is when that hyperpolarization happens. And the effect's determined by the receptors to which that it binds. So acetylcholine is excitatory in skeletal muscles, but it's inhibitory in cardiac muscles. When we look at the actions, which is direct versus indirect, direct is when the neurotransmitter binds directly to and opens the ion channels, and that promotes a very rapid response. And indirect is when it acts through intracellular secondary messengers. So it's much more broad, longer lasting effects, similar to how hormones work. We're gonna watch a little video about synapses here.
What's 1,000 times thinner than a piece of paper, more numerous in you than grains of sand on a beach, and proof that the smallest things can sometimes be the most powerful? I'm talking about the synapse, the meeting point between 2 neurons. If your neurons form the structure of your nervous system, then your synapses, the tiny communication links between them, are what turn that structure into an actual system. Because as great and powerful as your neurons are, when it comes down to it, their strength and their purpose lies in their connections. A single neuron in isolation might as well not exist if it doesn't have someone to listen to or talk to. The word synapse comes from the Greek for to clasp or join. It's basically a junction or a crossroads. When an action potential, and if you don't know what an action potential is, watch the last episode, sends an electrical message to the end of an axon, that message hits a synapse that then translates or converts it into a different type of signal and flings it over to another neuron. These connections are rather amazing feats of bioelectrical engineering, and they are also ridiculously, mind-bogglingly numerous. Consider that the human brain alone has 100 billion neurons, and each of those has 1,000 to 10,000 synapses. So you got somewhere between 100 to 1,000 trillion synapses in your brain. Each one of these hundreds of trillions of synapses is like a tiny computer all of its own, not only capable of running loads of different programs simultaneously, but also able to change and adapt in response to neuron firing patterns and either strengthen or weaken over time depending on how much they're used. Synapses are what allow you to learn and remember remember. They're also the root of many psychiatric disorders, and they're basically why illicit drugs and addictions to them exist. Pretty much everything in your experience, from euphoria to hunger to desire to fuzziness to confusion to boredom, is communicated by way of these signals sent by your body's own electrochemical messaging system. Hopefully, you know enough about email and texting etiquette to know that if you're gonna communicate effectively, you have to respect the sanctity of the group list. It's not a great idea to send a mass text to all of your friends first thing in the morning to give them the urgent news that you just ate a really delicious maple bacon donut. Seriously, people, if you happen to have a friend who truly adores bacon, then an email would suffice. But if you're out clubbing and suddenly Bill Murray shows up and starts doing karaoke, then that would be a totally appropriate time to notify all of your friends at once that something awesome is happening, and they better be a part of it. And in much the same way, okay, kind of the same way, your nerve cells have two main settings for communicating with each other, depending on how fast the news needs to travel. Some of your synapses are electrical. That would be like an immediate group text. Others are chemical synapses. They take more time to be received and read, but they're used more often and are much easier to control, sending signals to only certain recipients. Fortunately, your nervous system has better text etiquette than your mom and knows when each kind is appropriate to use and how to do it. Your super fast electrical synapses send an ion current flowing directly from the cytoplasm of one nerve cell to another through small windows called gap junctions. They're super fast because the signal is never converted from its pure electrical state to any other kind of signal the way it is in a chemical synapse. Instead, one cell and one synapse can trigger thousands of other cells that can all act in synchrony. Something similar happens in the muscle cells of your heart, where speed and team effort between cells is crucial. This seems like a good system, so why aren't all of our synapses electrical? It's largely a matter of control. With such a direct connection between cells, an action potential in one neuron will generate an action potential in the other cells across the synapse. That's great in places like your heart, because you definitely don't want half a heartbeat. But if every synapse in your body activated all of the neurons around it, your nervous system would basically always be in group text mode, with every muscle fiber and bit of organ tissue always being stimulated and then replying all to the whole group, which would stimulate them even more until everyone just got maxed out and exhausted and turn off their phones for good, which would be death. So that would be bad, which is partly why we have chemical synapses. They are much more abundant, but also slower, and they're more precise and selective in what messages they send where. Rather than raw electricity, these synapses use neurotransmitters, or chemical signals that diffuse across a synaptic gap to deliver their message. The main advantage chemical synapses have over electrical ones is that they can effectively convert the signal in steps, from electrical to chemical back to electrical, which allows for different ways to control that impulse. At the synapse, that signal can be modified, amplified, inhibited, or split, either immediately or over longer periods of time. This setup has two principal parts. The cell that's sending the signal is the presynaptic neuron, and it transmits through a knob-like structure called the presynaptic terminal, usually the axon terminal. This terminal holds a whole bunch of tiny synaptic vesicle sacs, each loaded with thousands of molecules of a given neurotransmitter. The receiving cell, meanwhile, is, yes, thankfully, the postsynaptic neuron, and it accepts the neurotransmitters in its receptor region, which is usually on the dendrite, or just on the cell body itself. And these two neurons communicate even though they never actually touch. Instead, there's a tiny gap called a synaptic cleft between them, less than 5 millionths of a centimeter apart. One thing to remember is that messages that travel via chemical synapses are technically not transmitted directly between neurons like they are in electrical synapses. Instead, there's a whole chemical event that involves the release, diffusion, and reception of neurotransmitters in order to transmit signals. And this all happens in a specific and important chain of events. When an action potential races along the axon of a neuron, activating sodium and potassium channels in a wave, it eventually comes down to the presynaptic terminal and activates the voltage-gated calcium channels there to open and release the calcium into the neuron's cytoplasm. This flow of positively charged calcium ions causes all those tiny synaptic vesicles to fuse with the cell membrane and purge their chemical messengers. And it's these neurotransmitters that act like couriers, diffusing across the synaptic gap and binding to receptor sites on the postsynaptic neuron. So the first neuron has managed to convert the electrical signal into a chemical one, but in order for it to become an action potential again in the receiving neuron, it has to be converted back to electrical. And that happens once a neurotransmitter binds to a receptor, because that's what causes the ion channels to open. And depending on which particular neurotransmitter binds to which receptor, the neuron might either get excited or inhibited inhibited, the neurotransmitter tells it what to do. Excitatory neurotransmitters depolarize the postsynaptic neuron by making the inside of it more positive and bringing it closer to its action potential threshold, making it more likely to fire that message onto the next neuron. But an inhibitory neurotransmitter hyperpolarizes the postsynaptic neuron by making the inside more negative, driving its charge down, away from its threshold. So not only does the message not get passed along, it's now even harder to excite that portion of the neuron. Keep in mind here, any region of a single neuron may have hundreds of synapses, each with different inhibitory or excitatory neurotransmitters. So the likelihood of that postsynaptic neuron developing an action potential depends on the sum of all of the excitations and inhibitions in that area. Now we have over 100 different kinds of naturally occurring neurotransmitters in our bodies that serve different functions. They help us move around and keep our vital organs humming along, amp us up, calm us down, make us hungry, sleepy, or more alert, or simply just make us feel good. But neurotransmitters don't stay bonded to their receptors for more than a few milliseconds. After they deliver their message, they just sort of pop back out and then either degrade or get recycled. Some kinds diffuse back across the synapse and are immediately reabsorbed by the sending neuron in a process called reuptake. Others are broken down by enzymes in the synaptic cleft or sent away from the synapse by diffusion. And this mechanism is what many drugs, both legal and illegal, so successfully exploit in order to create their desired effects. These drugs can either excite or inhibit the production, release, and reuptake of neurotransmitters, and sometimes they can simply mimic neurotransmitters, tricking the neuron into thinking it's getting a natural chemical signal when really it's anything but. Take cocaine, for example. Don't take cocaine. Once it hits your bloodstream, it targets 3 major neurotransmitters: serotonin, dopamine, and norepinephrine. Serotonin is mainly inhibitory and plays an important role in regulating mood, appetite, circadian rhythm, and sleep. Some antidepressants can help stabilize moods by stabilizing serotonin levels. And when you engage in pleasurable activities like hugging a loved one or having sex or eating a really great donut, your brain releases dopamine, which influences emotion and attention, but mostly just makes you feel awesome. Finally, norepinephrine amps you up by triggering your fight or flight response, increasing your heart rate, and priming muscles to engage, while an undersupply of the chemical can depress a mood. So in a normal, sober state, you got all these neurotransmitters doing their thing in your body, but once they've delivered their chemical payloads, they're usually diffused right back out across the synapse to be absorbed by the neuron that sent them. But cocaine blocks that reuptake, especially of dopamine, allowing these powerful chemicals to float around and accumulate, making the user feel euphoric for a time, but also paranoid and jittery. And because you have a limited supply of these neurotransmitters, and your body needs time to brew more, flooding your synapses like this eventually depletes your supply, making you feel terrible in a number of ways. Cocaine and other drugs that target neurotransmitters trick the brain, and after prolonged use may eventually cause it to adapt, as all of those synapses remember how great those extra chemicals feel. As a result, you actually start to lose receptors, so it takes even more dopamine and finally cocaine to function normally. Sometimes the best way to understand how your body works is to look at how things can go wrong, and when it comes to your synapses, that, my friends, is what wrong looks like. In their natural, healthy state, your synapses know when to excite, when to inhibit, when to use electricity, and when to dispatch the chemical messengers. Basically, a healthy nervous system has the etiquette of electrical messaging down to, well, a science. Today you learned how electrical synapses use ion currents over gap junctions to transmit neurological signals, and how chemical synapses turn electrical signals into chemical ones using neurotransmitters before converting them back to electrical signals again. And you learned how cocaine is a sterling example of how artificial imbalances in this electrochemical system can lead to dysfunctions of all kinds.
Okay, I hope that those videos were helpful to explain the things that we've been talking about. And then I will have at the end of the next video lecture the things to focus on for your quizzes. Again, make sure you're reading your book as well to get any additional information that maybe was missed in this lecture.